So I'm very happy to introduce Jasmijn, who will speak now about her project on algorithms to detect viral genomes in wastewater. So yeah, the floor is yours. Thank you for being here.
Thank you. Thank you for having me and inviting me to present here. So I realize it's quite a diverse group here, I think, in terms of technical expertise. I'm not sure to what extent you're familiar with what I'll be speaking about. So please interrupt me at any time if you have any questions, if anything is unclear. I'll try to not use too much jargon and terminology, but it might slip in, so please just let me know. I'm happy to talk or to answer any questions in between, and there should be plenty of time for that. So before I start, just quickly an idea of where I come from in terms of my background. So I actually studied here. I studied mathematics in Utrecht, where I focused mostly on algebra in my thesis. And then I went to Eindhoven to study applied and industrial mathematics, looking for a bit more applications. And I've also always been interested in biology, so for a PhD, I found a nice topic in Amsterdam working on bioinformatics. And from there on, I started studying viral genomics, reconstructing viral genomes, moved to Boston for a postdoc, also working on bacterial genomics, and I finally settled in Delft, where since 3 years now, I'm an assistant professor at the Intelligent Systems Department, focusing on algorithms for microbial genomics. That's a bit of where I come from. And one of the projects that I'm working on originates from the time that we were in a pandemic. I was in Boston, and people approached us with these wastewater samples, asking what can we get out of this? What can we learn in terms of the virus and what is happening, how is it spreading, etc. And wastewater is actually a very rich source of information. You can imagine it contains elements excreted from all people in a given geographical location. Everyone goes to the bathroom, so everyone contributes to this signal. And it can therefore be a very cheap and efficient way of monitoring public health and infectious disease, because you don't need to look at individual samples, but you can look at the whole population in a certain area together. This was already a field of study before the pandemic, but during the pandemic, this really took a big leap forwards, simply because it was very relevant and very urgent. And it became part of many surveillance initiatives. Already before, it was part of polio surveillance, but then, of course, we had SARS-CoV-2 surveillance. Now people are exploring many other infectious diseases, which can be monitored through wastewater, such as antimicrobial resistance also, but influenza and many other viruses. So how do we actually analyze genomes? Yes. Great.
Well, one question before you go further, because I was always very much impressed with this, and at the same time a little bit surprised, because we are talking about the respiratory virus. So how come it ends up in the wastewater? So I guess there is also, for different viruses or bacteria or whatever, there is a different rate of ending up in the wastewater, right? How do you deal with that? I mean, if you don't find something in wastewater, does it really mean that it's not in the population, or is it mean that it doesn't end up in the wastewater?
So these are very important questions, also quite tricky ones. Many of these people are still dealing with and trying to figure out what to do. And I think it depends on what you're looking for and the exact question you're asking. So if you know well what you're looking for and you don't find it, then I think you can be pretty sure it's not there. So sort of the presence absence thing is easier to answer than actually the amount of infections, for example. Like absolute numbers, that's very hard to estimate. I don't think we can actually do that reliably from wastewater data. Of course, there are things that people have come up with, like normalization and standardization approaches looking at very common viruses. For example, like tomato viruses. We all eat tomatoes, and these viruses like to live actually in our guts. And then this is sort of a baseline that you can compare to. But still, different patients might shed in different amounts. So overall, the data is very noisy. But if you take enough samples frequently enough, then you do see the bigger trends.
Yeah.
So there are different ways of looking at the content of the wastewater. And in this context, the two most frequently used approaches are either through PCR amplification or through genome sequencing. So you don't need to understand the exact details of how this works. But the idea for PCR amplification is that you know what you're looking for, and then you can try to amplify this specific sequence, so a target sequence here in red, by actually making use of the properties of DNA that you can duplicate it because it's double-stranded. So every base has a reverse complement, and you can use this property to build copies. And if you do this iteratively, you get many copies of the piece of genome that you're interested in. So say I'm looking for SARS-CoV-2 in a wastewater sample, and I know what the SARS-CoV-2 genome looks like, then I can design so-called primer sequences that bind to this fragment, and then I can amplify it. And doing many cycles of this amplification in the end results in many copies of this specific fragment. And then if in this process you incorporate some fluorescent dye, for example, that makes these fragments that you build let them send a fluorescent signal, then you can measure this and quantify how much of these fragments is in your sample. But it doesn't give you an idea of the diversity or the variation within those fragments. It only gives you sort of a quantitative measure of how much of this fragment is in there. So the other approach that is used often is genome sequencing, where actually you take the fragments and you sequence them. And this gives you short, fragmented DNA sequences, where we actually know the individual bases in the sample. And this we call sequencing reads. And
so the DNA you find in the wastewater is not of one thing anymore. It's already fragmented and it mixes everything.
Generally, yes. So you can still, I think, you can still find full genomes, but mostly they will already be fragmented into pieces because of all the other compounds in the wastewater that just damage everything, basically.
Yeah. Is it as soon as the cells die, the genome dies as well? Is it like that?
I think I don't think I'm qualified to answer this exact question. I'm actually
More to give an idea.
Yes. So of course, viruses are not living anyway, right? Yeah. They actually.
We're talking about viruses.
Yeah. So they are not living cells anyway.
Wrong question. Yes.
Yeah. So we can look at those fragments. And also, so what I want to say, if you had full genomes, we wouldn't be able to sequence those in one read anyway. We would always fragment them into smaller pieces and read those because those machines read smaller pieces. At least there's some nuance there, but for the most standard machines, they read shorter pieces. Now, genome sequencing obviously gives you much more information than doing the PCR amplification, but it's also much more expensive. So already early in the pandemic, people started looking at wastewater and trying to see, okay, can we indeed measure these concentrations of the virus material, and does it give us an indication of the number of infections? And actually, one of the first papers came from here in the Netherlands, showing that there is a strong correlation between the copies so basically the concentration in a wastewater sample and the number of infections in the population. And after that, many people have actually been showing the same thing, that we can track the number of infections by looking at concentration wastewater. And potentially even we can get an early warning signal, because in the wastewater, we see things before people get to the hospital and actually get sequenced, etc. So people can already shed the virus even before they have symptoms. So that's a nice aspect of working with wastewater data.
So that means that you already know what to look for. And how is it then an early warning signal?
Yes, so of course, if it depends on what you do, right? So if you have, you could look at the wastewater samples, just sequence everything that's in there and try to find something that you don't know what it is.
I thought that's too expensive. So you actually do that?
It doesn't, no, so that doesn't happen much, but it is one way that you could avoid having to know what is in there. In general, we do know what's in there, so you can do something less expensive and just search specifically for what you know is going to be there. Yes. Depends on what
you could have started looking for corona in January 2000.
I guess if there's an outbreak in China.
Yeah, exactly. Then you get the sequence and you can start
in
looking
this.
Yeah. So the difficulty with doing this is called metagenomic sequencing, where you don't amplify something specifically, but you sequence just everything that's there, is that you're really looking for a needle in a haystack. So you will only be able to find it if it's sufficiently abundant. If you know what you're looking for, then you find it earlier. Yes. So of course, as we've seen during the pandemic, the virus keeps evolving, right? So it accumulates mutations in its genome, meaning that some positions change. These mutations are used to define so-called lineages of the genome. So these are slightly different variants, or like slightly different versions of the virus. So they have a slightly different genome. And then we use the term variant for groups of lineages with similar mutations. So for example, we see the alpha variant, the delta variant, Omicron, etc. And this plot here comes from NextStrain, which is a platform used to show the virus evolution and all the data that we have for this. But this is based on clinical sequencing, so meaning that individual patients coming to the hospital, they have been sequenced to see what exact lineage was infecting them, and that gives you this plot showing how the abundance of these different lineages has changed over time. But obviously this is very expensive. It's time consuming because you have to analyze many samples. And it's also biased towards patients that are very sick and come to hospital. Right? You don't see the ones that don't have any symptoms or barely any. So then the question was, what can we actually look at the wastewater and still distinguish between these different variants or different lineages? So can we track them over time from our wastewater samples? Now, as already came up just now, is that these samples are actually very noisy. So first of all, we have to deal with the stochasticity of sampling. So depending on the time of the day, you might get different concentrations and different contents of your sample. Then in the sample itself, the genomes degrade quickly. So the material that we get is actually of quite low quality and also of low concentration. So most approaches amplify the genome, like the PCR amplification that I showed, before they sequence it. So they first amplify what they're looking for and then sequence those fragments. But this in itself is difficult because there are many compounds in the wastewater that inhibit these reactions that are needed. And finally, in doing this amplification, you get all sorts of biases. So in general, the data is tricky to work with, and to give you an idea of what it looks like, I have some plots here that show you for each position in the genome. So on the x-axis, we have the position in the genome, and on the y-axis, how many fragments we see for this position. So this is a very low quality sample where in most positions, we don't get any fragments. We only get it for a few, you see a few peaks where we do get measurements. Then this is a slightly better version, or like a slightly better sample, where we actually, for 61% of the genome, we get coverage. So meaning we see at least 100 reads from this position.
Is the ideal situation just uniform?
That would be perfect, yes.
That's what you're sort of expecting theoretically, right?
Yes, but because of all these issues and all these biases, that's never what we see. So even for one of our best samples, where we have 97% of the genome covered, you still see it's very spiky and non-uniform at all.
And are the fragments they use for the PCR somehow correlated to the parts which are very present in here, or is it just kind of purely based on other characteristics?
So it has to do with the PCR also. If I understand correctly, like the important aspect is the primers that you need for the PCR, so the sequences that need to bind to the fragments in your sample in order to be able to amplify the part in between. How well they match, for example, to the template, so the part that you're trying to copy.
Is it to be 100%, or is there some?
There can be some differences there. They don't have to match for 100%.
I'm really wondering, if you only amplify what you know, how can you find variance?
So I'll wait with answering that until a bit later in the presentation. I hope it will become more clear then. But for now, I hope this gives an idea of what we're actually working with. So where to start? Many people were looking at this in the pandemic, and the first, most sort of naive thing we do is to take the genome that we know, so we call that a reference genome. It's here on top, illustrated by just a line, and we have our fragments, and we align them to the reference genome. So we see where in the reference genome does this fragment belong, and do we see any differences compared to the reference genome? For example, you might see this blue mutation, so where a nucleotide has changed at a certain frequency. We see a purple one and a yellow one, and they will appear at different frequencies. So now suppose that we actually are looking for 3 different lineages, and we know what these look like, how these are defined. Namely, lineage 1, for example, has the blue and the yellow mutation. Lineage 2 has blue and red, and lineage 3 has blue and purple. I realize the colors are not very clear, but I hope the general idea is clear, that then based on the frequencies that we observe, we can figure out how abundant the different lineages are. For example, only the lineage 3 has this purple one, so the one in the middle, which appears at 70%. So then this lineage must make up 17% of our sample. So this is very ad hoc, but there's also a clear downside of doing this, because you are limited to looking at lineages with these unique signature mutations. So for example, the purple one, which is clearly only for this lineage, or the yellow one only for lineage 1, etc. So then a more sophisticated approach is to actually make a matrix where you record your lineage defining mutations. So for each lineage, which mutations belong to this lineage, and you have a vector of your observed frequencies of each of these mutations. And then what we can look for is a vector X that has the predicted frequency of each of the lineages, and ideally A times X equals this observed vector B. So you're trying to minimize this absolute deviations problem. And what people then have done is to actually incorporate some extra information, namely the amount of reads we see for each position. That's called a sequencing depth. So the more information you have for a position, the more important it becomes in your problem, because you have more information there. So you can make it a weighted absolute deviation problem. So that's one approach to do this a bit more sophisticated. But this still assumes that we know these frequencies of the mutations in our sample. And in reality, measuring these frequencies is very error prone, because it's quite difficult and it requires high sequencing depth. So you need a lot of data to do this accurately. It's based on placing your reads on the reference genome, so figuring out where they belong, which means you always have to deal with bias towards this reference genome. And this approach does not actually consider combinations of mutations in predicting the frequencies. So if you know that a combination of mutations belongs to a certain lineage, then you might want to look at fragments that have this combination together in them. But common approaches in quantifying the different mutation frequencies do not consider this. They look per position. And finally, if you think back to what the samples actually look like, where in many places we don't have any information, then if your mutation that you need to quantify your lineage is in one of these positions, then you lose all information. So that's why I say it's susceptible to amplicon dropout, meaning we don't have information.
Can I ask something?
Yes.
This they have here that you only have not use this combination of SNPs. Is that a problem? Because do these SNPs often occur in the same kind of read, or if they're kind of further apart in the genome, of course, then it kind of becomes independent anyway.
Yeah. So with high mutation frequencies, then you would expect them to appear together. And it simply gives you more information because there's mainly because the sequencing machines themselves also make errors. So if you see the same mutation or like a combination of mutations appearing in multiple reads, then you can be more certain that these are not errors from the sequencing machine, but that it's actually a true signal. So in that sense, it gives you more information and you would be able to do things more accurately.
But if you're using first generation sequencing, you're getting very short reads. So if these mutations are even some distance apart, you wouldn't see them in the same read, right?
So with first generation, usually we would refer to Sanger sequencing. Like the technology I'm referring to here, which is most commonly used now, is second generation sequencing, which is like Illumina.
Yeah, I mean, even in that, you would get pretty short reads, right? I mean,
Yeah, now you have, of course, the possibility to have longer reads. So I guess that will solve some of the problems, but I guess that's not being used yet, or
not much. In wastewater, it's tricky because your sample quality is already so low, and you need to do amplification, and you need to
But even with the short reads, you don't necessarily need to know that the reads come from the exact same fragment. As long as you can assign them to a certain lineage or a certain variant, you can look at the differences in the quantities of reads assigned to a certain lineage versus another. And that is still, it's noisy, but it's a quantitative measure of the presence of different lineages in your sample. You're not interested in the individual virus, you're interested in the whole population. So even if you have small variations, there's algorithms you use to correct these things and cluster your reads such that you end up with a number of how many reads this particular lineage has, right?
Okay. Yeah, so it's not a must that you can do this. It's more like a bonus. If this information is there, you would want to use it, right? Yeah, so in general, this is the drawback of doing something that's based on mutation frequencies. And therefore, we considered another type of approach where we do not depend on this. And this is based on read-based expectation maximization. So I'll show you what I mean with that. And so there are many approaches that have that attempt to do something with wastewater data since like the pandemic started. Most of them focus on this SNP-based deconvolution, but similar to us, also Baaijens et al. Came with something based on read-based maximization. So I'll tell you a bit about what we thought of. It is actually a very simple idea based on what was already done before. Namely realizing that the problem that we're trying to solve is very similar to the problem of RNA transcript quantification, where you know a sequence for a gene, and you know the different transcripts that can appear for this gene. So you have a collection of sequences that you're interested in, and you have sequencing data for the sequences in your sample, and somehow you want to decide which sequences belong to which, so which of the sequencing reads belong to which of the genomes you're looking or interested in, and then predict the relative abundances of these sequences. And this is a well-studied problem and had many methods to do so. So our idea was, okay, if what we're trying to do is so similar, maybe we can actually just borrow the techniques that they use to solve our problem. So we went along with Callisto, but it's very similar to the tool Salmon actually, and we tried both, but found that Callisto worked slightly more convenient and more accurate. So the workflow for this is that you start with your collection of genomes that you're interested in, so your reference set. And you build an index for this, which is basically what we call a colored line graph. So it's a graph that records these sequences, and it shows you where they're the same or where they are different. So in that sense, you see that the paths branch when they're different. They come together again when the sequences are the same. And then when we have our sequencing reads, we align them in an efficient way. So it's called pseudo alignments to this graph. So we know which paths in the graph they follow, and based on that, for every read, you can decide to which of the paths it is compatible. And based on that, then with an expectation maximization step, we quantify how abundant each of the genomes that we're looking for is. And this actually works. So we did some experiments. It worked quite well. But the difficult part is that you need a set of reference genomes to start with. So you need this set of initial sequences that you're going to be looking for. And we have a lot of sequences for SARS-CoV-2. We have this GISAID database, which has, in the meantime, millions of sequences. So how do you select which ones to use? How do you know what makes a good reference set to then estimate the abundance of these sequences? So in our pipeline, which we called VLQ, we do a very simple and naive approach where we just look per lineage at all the sequences we have. We check which mutations are there and how abundant they are, and then we include anything with a frequency above 50%. So anything that seems important to this lineage. So you may realize that this, again, it does use mutation frequencies. We do need to identify them, but only for selecting our sequences. From there on, the estimation itself does not actually depend on this process. So then for each lineage, we select some sequences that we predict the abundance for, and then we can sum those together into the classes that we're interested in. So suppose you want to report abundance for B117, then we sum all the sequences that we have for B117 and sum this into a final predicted abundance. So this is a simple approach which worked, but then the question is, okay, even for this selection, do you want to use your entire database, or do you want to be more selective already in your input to this selection process? Or maybe there are better ways to do the selection process. Probably there are. But what we saw is that actually the way you select your reference sequences, it impacts the prediction accuracy. So this is actually where master student Johanna and my PhD student Jasper, they started working on this problem, trying to see, okay, how does the way that we select our reference sequences, how does it impact our prediction accuracy? And one of the things we looked at was, as starting points, taking sequences only from a certain geographic region, like different skills. So if the data set we're looking at is in a specific state in the US, for example, we can take sequences only from this state as our reference set, or we can take from the entire country, from the entire continent, or just all sequences globally. And what we see, and I think intuitively it also makes sense, that the more local you go and the more specific your sequences are, the better you can actually estimate. So the errors go down. Where the different lines in each of the plots are different ways of clustering sequences and then selecting a representative sequence. So we're still working on this also in different contexts to really understand what makes a good reference. My main point now with this figure is to actually show that what we see is that the selection algorithm and the sequence locality, so the region you select from, they have a substantial impact, and it is something to consider when you choose your reference sequences. And then in doing these experiments, there was another observation, namely that selective amplification can improve our prediction accuracy. So now we're also coming back to the parts that, well, to some of the discussion earlier, where we have to amplify the material in our sample, or at least this is the standard approach. Because we're looking for a needle in a haystack and concentrations can be low, so what people do is they first use this PCR to amplify the material that we're looking for. And the standard approach is to have this scheme of amplicons, it's called tiled amplicon, tiled amplicon design. So we have shorter sequences which together cover the whole genome of interest, and each of these shorter sequences we amplify to get enough data to analyze. And there are different ways of doing this. But another approach could be to just look at a region where you have most information. For example, for SARS-CoV-2, most variation occurs in the spike gene, which is here slightly towards the end of the genome. So you could only look at amplicons in the spike gene, or even just look at a single amplicon. If this amplicon has most of the variation, it might already allow you to distinguish between lineages and quantify them. And in our simple benchmark experiments for this quantification approach that we developed, we compared also to doing whole genome sequencing, so having reads for the whole genome, which is the figures on the left, versus having only reads for the spike region, which are the figures on the right. And what we saw is that in general, we get lower errors if we only look at the spike region. So that made us wonder, okay, if most of the information is there anyway, and this gives us better results, then how do we select the best part of the genome to sequence? So the initial question that we started with was, how can we track the lineage frequencies over time? And then we saw, okay, it matters which reference sequences you use, so which ones should we select? And a third problem that came up was which amplicons give us the best abundance estimation accuracy? So you see, you start with one question and you get many in return. But this means that we still have work to do. And in terms of the third question, we actually came up with an approach to solve this. So just to make sure we're all on the same page, for PCR amplification, what we need is primer sequences and a known target sequence. So suppose that this is a part of a piece of DNA of the genome we're interested in. What PCR will do is you have your primers, so here are the gray sequences that bind on either side of the DNA, and then you can amplify the piece that follows it. And by doing this iteratively, you get many copies of your sequence. But we have requirements for these sequences. So the primer sequences, the gray ones, they must bind to the sequences that we're interested in. They should not be too long, so say between 18 and 28 nucleotides. And they need to satisfy a bunch of chemical properties like melting temperature, well, many things that I won't go into now. And then for the amplicons, so that the part in between that we're actually interested in, these are the regions of interest, and we want to select them such that they allow us to distinguish between lineages. And again, to have these primers, the regions surrounding the amplicons must be conserved, otherwise our primers won't bind to all the genomes. So these are two important terms to memorize. Yes?
How long are these amplicons compared to the primers?
Depends on your design. Basically, you're free to do whatever you like, but anything in general for PCR amplification above 1,000 bases or so, it becomes trickier to amplify, not impossible, but more difficult.
So the pictures are not the size, definitely.
But for wastewater samples, 1,000 is already quite long because your fragments might not be that long. So the standard schemes, they use amplicons of about 400 base pairs. Okay. So what then Jasper, my PhD students, start looking at is, okay, if we have this database of all those sequences, and we would take an amplicon of a certain size, so here we consider either 200 or 400 base pairs, starting at a certain position, between how many pairs of sequences, how many pairs of lineages can we distinguish based on just this sequence? And then the relative differentiability is the relative amount of sequences compared to all pairs of sequences, how many pairs relatively can we distinguish between. So you see that for certain regions of the genome, there's much more information than for other regions. So here you see, my pointer of course doesn't work, but there's the spike gene, S1, S2. There's a lot of information there, but actually most information is in this final gene, the N region. And what we want to do now is to select amplicons along with primers. So we need these conserved regions around them, and make combinations that give us most information. Because you could select, for example, your first amplicon in this highest peak in N, but then for the second amplicon that you select, you want to take into account the information that you already extracted from this first amplicon. Right? So you don't just go for the second highest peak, you actually want to make the best choice, taking into account the earlier information. So the goal is to select amplicons and primers to optimally distinguish between different lineages. And to do so, we actually formulated this as a set cover problem, but also realized that it's hard to solve. So we resorted to a simple greedy algorithm to come to a solution relatively quickly, where the final outputs are the amplicons along with their primers. So in the interest of time, I won't go into the details too much, but just to give an overview of what we do. We start with our database on top of all the sequences, and we make a multiple sequence alignment. So that's our starting point to know, okay, which regions in which sequence align, so which correspond to the same thing. And from our set of references, we also build a primer database. So which possible primers could we use in those sequences? And then using the multiple sequence alignment, we generate candidate amplicons. So these amplicons need to satisfy some requirements. They cannot differ too much in length. I think that's the main requirement there to make an amplicon actually feasible. So we get this set of possible amplicons, and then we enter this iterative cycle where we select the best amplicon in every step. So for the set of amplicons, we rank them by their differentiability, so between how many pairs of sequences can they distinguish. And they should be new pairs of sequences, so taking into account excluding the sequences that we've already been able to distinguish between. Then we take the best one, check if we can find primers for it. If yes, we add it. If not, we throw it out and we try again. And this we iterate until we're happy with the number of amplicons we found, or we are satisfied with the differentiability that we have reached. And then looking at what we actually get, of course, our first amplicon is going to be in this highest peak in N. But after that, we'll see that the amplicons are quite nicely spread over the genome. And here the heights of where the amplicons are indicate the additional differentiability that we get from this amplicon by adding it on top of the previous amplicons. So the next plot actually shows that better, where we see the cumulative relative differentiability for amplicons with 200 or 400. And you see that, well, of course, larger amplicons is better, but it nicely increases and converges. So already 3 amplicons is actually sufficient to reach 86% differentiability if you take amplicons of length 400. So you only need a few amplicons to already extract most of the information from your sample. So then finally, we wanted to see also how this then impacts our prediction accuracy. So if we simulate sequences from just those amplicons, and we then try to predict relative abundance from this compared to having the whole genome, what happens to our prediction accuracy? And it depends a bit on the data set. So we had 2 data sets here, one representing sequences from Texas, the other representing sequences from the Netherlands. And for the Texas data, you can see that actually using our selected amplicons, we reach a similar accuracy to whole genome sequencing. Again, more amplicons is better, but even with a few amplicons, we do quite well. For the Netherlands, we see a different story, where whole genome sequencing clearly does better, but there are some considerations here. The first is to realize that this is not actually a fair comparison, because in our amplicon data, we check if the primers that we've selected actually bind to the sequences that we should be simulating from. If they don't bind, we also don't simulate. So we don't get the data. So our data for amplicon sequencing is much more realistic compared to the whole genome sequencing, which is also based on this tiled amplicon scheme, but there's no check if primers actually bind to the sequences. So this is the whole genome setting is far idealized and in no way a fair comparison. But what we also see is that in the Netherlands data set, there are many Omicron sublineages with some inconsistent naming. And if you would actually quantify on a slightly higher level, so for example, not going into BA 2.5.8 or whatever, but just BA 2 point something, then those differences disappear. So if you don't try to be as precise as we do initially, then with amplicons you are actually on a par, even in this unfair setting with whole genome sequencing. Okay. So to wrap up, what I've shown you is that we can use wastewater sequencing data to analyze, well, basically to do genomic surveillance and to see how a virus is evolving over time. Individual samples in this are very unreliable because they're like simply because of the nature of wastewater. But we can track variant or lineage abundance over time. And I showed you this approach that we use for this, where I want you to remember that reference set composition, so the sequence that you put into it, this is really important and it strongly affects your results in the end. The second thing to remember is that when you design your experiments, sometimes looking only at the really most informative piece of information, so in this case selective amplification, can be a very cost-efficient alternative to looking at all your data, in this case whole genome amplification. So then to really conclude, we can use wastewater-based surveillance as an early warning system. Of course, there are many things to consider and issues to be solved yet, but it could be very beneficial, especially in low resource settings where there are no facilities to actually do patient sample sequencing, or there's simply not enough money to do so. And in terms of computational challenges, what we do need is to have more robust pipelines, also to be able to discover novel sequences. So if we don't know what we're looking for, we need to have proper reference databases, where actually this is a database that I've been referring to. In the meantime, people have shown that sequences in there are highly contaminated. There's no proper quality control on these. So we need to have a better system for this, and also make sure that approaches are sufficiently scalable. Of course, there's also experimental challenges to deal with all those biases that I mentioned, and ethical challenges. So if you look at wastewater samples, to what extent, who owns the data, right? It's what happens to privacy concerns? How can we develop policy based on this? So there's many things to consider in this context. But for the things that I'm working on, the most urgent now would be to have actually proper benchmarking, realistic scenarios, and also experimental validation of for example, the amplicon design algorithms that we've developed. If you want to know more, please have a look at our papers on this. And with that, I would like to thank all the people involved in this, in particular my students, Joanna and Jasper. And of course, thank you for being here and listening.
What is a realistic number of amplicons to have for a data set? Like, is there an upper limit to it, or for all intents and purposes, you can have 10 if you please, and that's fine. It doesn't really affect the sequencing experiments.
So, I mean, let me just show the standard approach again. It's a little bit there. Yes, here. So the standard approach is to have 30 amplicons or so that cover the entire genome.
Hmm.
So there's not really a strict upper limit, because you can just do all. But of course, the fewer, the less resources you need. Yeah.
And the better you can allocate your total bits. That's what I was thinking about. Okay. Yeah, that's interesting. Yeah, I know. I have worked with 6NS before. Like, one of the points of contention is whether to really go all out to one single amplicon or a couple of amplicons, or try to cover the whole gene. And well, there's people in both camps. Okay.
It depends on the question you want to answer, also, I think, and the data that you have. So yeah.
No, of course, here the interesting thing is that, because I suppose that you are not focusing so much on everything you can find in a sample, and rather on a virus of interest. It's a different way of looking at the problem. And what about, if you haven't worked with this, it's completely fine, but I'm curious, is there a significant difference with when you're looking at a DNA virus versus an RNA virus? Because I imagine, especially for obtaining the reference genome for an RNA virus, because of the fickleness of RNA in nature, it's probably quite difficult to get that a good quality reference genome, right?
So I think the main difference between RNA viruses and DNA viruses from this perspective is the mutation rate. So RNA viruses evolve much faster because they are single-stranded, so they don't have all these error corrections or all these mechanisms that allow them to sort of protect themselves. So they change faster, so you will more quickly have a problem with reference bias or reference not representing your sample sufficiently well. But in this case, like if you have patient samples or if you have other sources to actually get your enough data to assemble a good reference genome, then I don't think it really is an issue at this point.
Yeah, because besides the mutation rate, I'm also thinking directly about sampling, because RNA is so much more unstable than DNA that, like, even if you ignore the problem of the mutation rate, when you sample, it's probably already completely scrambled and, well, okay.
Yeah, so for DNA, you might be able to get higher quality samples, that's what you mean, yes. Also, like with RNA, usually it's first converted to DNA, which adds another step in the process that can introduce errors and biases. So I guess you're right, and that working with DNA viruses would probably be a bit easier. But I haven't done so.
Okay.
I was a bit surprised when you showed the data that you have data kind of only selects strains which are present in the area, so basically state level in the US, but it does so much better. Because intuitively, I would expect that if you add something which is just not present in the data, we never follow that part in the brand graph, so it will be kind of eliminated anyway. So what is the reason why it does so much better?
I think because sequences can be very similar, so it's very hard to distinguish between them. And then if you add more sequences that sort of confuse the process, then it gets harder. But it depends, of course, on what you're adding and what's in your sample.
Because if you would add, let's say, not a SARS-CoV-2 virus, but let's say an HIV virus, then of course it doesn't do anything.
No.
So I was
That's what I think, yeah.
That's to do with the fact that there's so much error everywhere that you end up at the path of other kind of virus, although it's just not there, but just because you happen to have the single mutation, it's kind of single mutation, single sequence error that you still go on that path. Something like that.
And possibly also for regions which are very conserved, so they actually are the same between many of the genomes. They could be assigned to any of them, and then often you see that they all get a little bit.
They're not assigned proportionally to what you find at the other places.
Ideally, yes, but in practice, not perfectly, right? So of course, there's region in terms of there's room for improvement in terms of the quantification step as well, I guess, to solve it. But yeah, it's, I guess, all three aspects matter, right? So what are your reference sequences? What is the region that you're actually looking at? And then how do you do the quantification? And in all parts you can improve. And they all affect the final accuracy.
Oh, you go ahead.
I was wondering about the benchmarking. Do I understand correctly that those are based on simulated sequences? It's not that you actually performed the experiment with sequencing with the primers that you.
What I showed was indeed based on simulated data, simply because then you know for sure what's in there. So you have a ground truth that you can compare against. For the quantification part, we also did a lot on real samples, but it turned out very tricky because then you can compare to the clinical case numbers, but like you cannot even expect these numbers to match perfectly because you're measuring a different thing at a different time. For the amplicon selection part, we're now actually working on experimental validation. So we do know that our primers work. Okay, we get something there, yes.
Okay.
But we're not to the part yet where we've actually then sequenced and ran our quantification on that to see the actual differences, because that in the end is going to be the most informative thing to, like, yeah, I think very valuable to have.
Yeah, I agree, because I was wondering, in your algorithm for this the design of the primers, do you use an external tool? Because there are many tools to design primers, which is quite tricky, as you already mentioned.
Yeah, so we don't, because none of them satisfy the requirements that we have, because we also allow for degenerate bases. So that means like bases which are not uniquely defined, but which can be an A or a T, for example. And the primer tools that we looked at, like, so I mean, this was mainly Jasper exploring all the different tools out there, and all of them had some reason why they could not work in our context. So yeah, it's our own check of all the requirements based on these tools. So based on what they also check, we check the same things. But yeah, in the end, experimental validation will show that it actually works. And I think PCR is a mysterious thing that even people doing it like don't always understand why it works or why it doesn't work. So yeah, we'll have to see.
Yes. I just wanted to ask how much of this data is actually publicly available? So I guess RIVM in Netherlands is collecting this data, but can you analyze it without a problem, or?
You mean for wastewater samples?
Yeah,
for the
wastewater
genomes?
samples, yeah.
Wastewater samples. Yes, so this runs in the Netherlands. It's being run through the RIVM, and I think much of their data is available. Probably not all. I haven't actually looked into it because when I started this, I was in the US. So we were working with US partners and looking at US data sets. And since continuing this in the Netherlands, we've mostly worked with simulated data for the reasons I just explained. So I haven't actually looked at RIVM samples, but I think at least for like they've published also things on this, so those data sets are available. I don't know if all is available. I'm not sure.
Thank you.